From Microbe TV, this is Immune, episode number 25, recorded on October 30th, 2019. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hello again.
I bet it's cloudy up there, right?
It's cloudy,
Yeah,
but it's
same
been
here.
beautiful, so it's not too hot.
It's been interesting weather, not at all cold, right?
No, it's been really warm.
Also joining us from Durham, North Carolina, Steph Langel.
Hey there. Good to be back. Episode number 25.
It's
good to hear your voice.
A quarter century of Immune.
I know.
Well,
We're progressing.
Not quite.
I know. Not quite.
Well, if you like Immune, consider supporting us financially. We could use your help. Microbe.tv/contribute. I will tell you more about that later. So stay tuned. And also, we're going to give a book away. Is this our first book giveaway on Immune?
No, we did one more.
We did another one?
Yeah, there was one more.
I don't remember which book it was, but yeah, we gave one away.
All right, you're going to want to stay for this one. And, well, I don't know when I'll mention it, so you just have to listen.
Anticipation.
But, you can learn. And today I learned a lot. I picked a paper, which I say is out of my comfort zone. I initially said there are no viruses in sight, but there happens to be. But it's really just kind of as a tool.
It is.
Yeah,
it is.
I don't really care about the virus. Just we have sequences that we know about. So, but it is really very interesting, and I like it because it's a nice example of where the microbiome is maturing, and that we're starting to do really functional experiments. Instead of saying the microbiome is important for everything, we're saying here's a specific example of how it's important. So we talk a lot about microbiomes on TWIM, and Elio is always mad because he said, this doesn't mean anything. Where's the mechanism?
Yeah, it's nice to work into some mechanisms, not just look at gene expression or just population differences. Yeah, it's a good paper you picked. Even though there's a couple of virus mentions. But as you said, everything goes back to viruses.
Well, I had some other papers that were very viral, and Amy, my lab here, she said, you should do it. It's a really good paper. And so
it is a good paper.
It's published in Immunity, and it's called the Microbiota Derived Short-Chain Fatty Acids Promote the Memory Potential of Antigen-Activated CD8 Positive T Cells. And the authors are from a variety of places, mainly in Australia, University of Melbourne, which I've been to, University of Würzburg in Germany, which I've been to, the Walter and Eliza Hall Institute for Medical Research, which I've been to in Australia, the Max Planck Institute for Infection Biology in Berlin, I have not been there. James Cook University in Australia, I have not been there. Monash University and Peter MacCallum Cancer Center, both in Australia, I have not been to. Who cares what I've been to, right? Why do I mention that?
It's interesting.
Well, I thought when I got through the first three, I said, wow, am I going to have visited everywhere? But no. And the first author is Annabelle.
You're checking off your list.
Annabelle. So the last name, I could pronounce a number of ways. Bachem, Bachem, Bachem. I think there's a company called Bachem. Annabelle, that's cool. Annabelle is a cool first name.
I like that name, yeah.
And then there's lots of others. And then the last two are co-authors, Thomas Gebhardt and Sami Bedoui. Or Bedoui. Could be does anyone know any of these people?
No.
No.
I don't either.
If they hear this, they can write in.
So my two hosts here, please jump in any time to correct or augment. I really should not do this, right? Or maybe I should, because
no, you should.
I should do it. So it's like doing a journal club.
Yeah.
The thing is, I don't have the insights that you two will, so I can have insights in virology, but I can ask questions. Anyway.
But these are T cells, right? And
they are T cells.
Steph, you're a B cell person. I'm a macrophage person. So
okay, well, I'm sure
we'll do the best we can.
We will each bring in a couple little tidbits here or there. I think we'll make.
So
this
is about
cool.
CD8 positive T cells. Now, so those are ones which can become cytotoxic T lymphocytes and kill virus-infected cells, and they can also make cytokines and do things. So these get instructed by antigen-presenting cells. So if you have an infection, the antigen-presenting cells like the macrophages and dendritic cells, they will present peptides to these CD8 positive T cells, and then they can become effectors and do things, or they can become memory to give you long-lasting memory, right? So that if you get another infection, then these T cells will come back quicker. And so that's what this is about, the process leading to memory, which I know virtually nothing about. I just know it's there. We have memory of all kinds, and this one is CD8 positive memory. And this is very well studied, of course, because people want to know what contributes to good memory and bad memory, especially with vaccines and other antigens. But many people study. We know that there are distinct populations of RNAs, mRNAs associated with memory, epigenetic changes as well, modification of chromatin and so forth. But changes in metabolism is also involved. Mm-hmm. And in this paper, they say, well, these cells, these T cells, once they get instructed, they traffic through other tissues, and they may get cues from those tissues as well. And of course, there's this whole microbiome that we have, and there must be some role for that. And we know for CD4 positive T cells, there have been a number of links made between microbiome and memory. And they cite studies using germ-free mice. So we're going to use two kinds of mice in this paper. One of them is germ-free, which, as the name says, which I hate. I don't like germ, but that's the name we're stuck with.
You don't like the word germ.
It's kind of germs and bugs, I'm not really fond, but fine. That's the name we stuck. They don't have any germs in them. They're clean mice.
Right. All right. And
previous studies have shown that short chain fatty acids, these are little chemicals derived from fermentation of fiber. So in fact, when you have a high fiber diet, this is one of the benefits. You make these short chain fatty acids, which can be beneficial. And these are metabolized by the bacteria in your gut. You eat fiber, goes in there. We can't handle fiber, but our microbiome can deal with it, and they can ferment it to produce these short-chain fatty acids, which include butyric acid or butyrate, acetic acid or acetate, propionic acid or propionate. And these get absorbed. They're short-chain fatty acids. They can go into the epithelium, and they can have effects either locally or at distances. And others have shown that these are involved in T-cell maturation. So here they wanted to say, what is the role of this microbiome and these short-chain fatty acids in the production of CD8 T-cell memory,
right? Specifically.
All right. So that's what we're going to look at. So they have these germ-free mice, and then they have specific pathogen-free mice, or SPF mice, which, as the name says, they lack certain microbes, but not all of them. Right?
Right.
And you can get a list, say they're missing these, but so they're not germ-free. They have some.
Right. They have a microbiota in their gut. They're colonized, but they're not going to have things that either make them sick or otherwise would affect your experiment.
So if you bought,
and it can be different from facility to facility, right? Because some facilities are very strict, and you have to rederive the animals into the facility to make sure that they don't have any of these mouse pathogens. And then other ones they kind of have accepted pathogens. So certain ones they allow and certain ones they don't. So there's some, like everybody has usually CMV or a herpes virus, right? So it would be like that, allowing some of the things into the facility and not others. So it's kind of weird because we call it specific pathogen free, but they have some pathogens. And then there's this idea that the less pathogens we have, the less mature the immune system is, right? And so that affects the interpretation of immunology experiments. And people are going the opposite direction now to have them with wild mice with all the different parasites and pathogens that they're normally exposed to and looking at what happens to immune responses in that context, which they argue is more relevant to what a human would see because we're walking around bathing ourselves in lots of different microbes. And so our immune responses in the context of all that other stuff going on are what's really relevant. So it's interesting how you can use, mice that have different levels of colonization to ask this, but they chose these two because they're pretty standard.
So if you can buy SPF mice, right? And then you put them in your facility, but they may not remain SPF in your facility depending on how clean it is, right?
That's true.
I did an interview with Andrew Gewirtz at Georgia State, and this will be out in months. And he has
Georgia Tech.
No, Georgia State.
Oh, he just published.
Yeah, so he published this story where they infect mice with rotavirus. So they use rag knockout mice, and they get chronically infected with rotavirus. And they were breeding these rag knockout mice in his facility, and all of a sudden they became resistant. Long story short, they were colonized by segmented filamentous bacteria, which made them resistant to rotavirus.
Yeah,
which they had to tease out, and they acquired it in their facility.
Yep.
Oh,
it's very interesting.
It is the way that
very
came about.
Anyway, so we have two mice, specific pathogen-free, SPF and germ-free mice. So then they give them, which we call adoptive transfer, T cells, a very specific line of T cells that has a T cell receptor that is specific for a glycoprotein peptide from a herpes simplex virus. All right, so it's a short peptide which is recognized by this T cell receptor, and they do that so that there's no need to select certain T cells in the animal and so forth, so you know exactly what you're getting, right? So these are cells, T cells. They add, they're specific for this herpes simplex peptide. These are called GBT1 cells. I'm going to use these throughout the paper, in fact, okay? So they give these to the mice, the two kinds of mice, and then they ask after transfer 7 and 14 days later how many cells are there, and they notice that the germ-free mice, there are fewer cells in the spleens on day 7 and even fewer on day 14. So there's a decline in these cells that they've injected into the mice initially, and so they say, if it's germ-free versus SPF, it must have something to do with the microbiome, right?
And I want to ask a question to Steph at this point. You've done probably more flow cytometry than I have, but
sure.
Their differences are 0.07% compared to 0.05%.
Yeah,
I mean that's data figure 1 that you see, and my first reaction was, what the heck? I mean,
yeah, so this is definitely a criticism that can be put on flow cytometry experiments, especially in these type of mice studies where when you look at the stats, they say the N is always greater than 3, but I believe it says could even equal 3. So you're maybe talking about 3 or 5 mice. And what they are describing, these are rare populations of cells that they have adoptively transferred and have migrated to the spleen. So to get a significant difference of what I'm looking at B, there's 3 stars. So, I mean, that's highly significant. But look at the numbers. And I think when you see flow data on rare cell types, there have to be more functional studies, which I think I could have used more of in these early experiments.
Me too. I mean, so what they did here was just so you guys, are clear who are listening, is they use a marker, this CD45.1 that I think we've talked about in a previous episode, marking different subsets that you put in or mix together, and you can identify them by this marker. And so they look at this when 7 days after transfer, and these were in vitro activated cells, so that's important too, because then they're looking for memory later. But so they put them in, and they're measuring 0.05 or 0.07%. You move that little gate just a fraction, and it completely changes the numbers. And so then they just multiply that by the total number of spleen cells. So if you multiply 0.07 by a million and 0.05 by a million, you end up with pretty big differences if you graph it.
Yes.
So
you're really expanding the magnitude.
Yeah, these are, a lot of places in this paper, there are really minor differences.
Mm-hmm.
And so I think really worry about some of the data. I mean, functionally, yes. And when they get to the metabolomics, I think it's pretty clear, although I can't evaluate some of that because, they just have some graphs and I don't know what makes up that data. But these differences are extraordinarily small.
Yeah.
Yeah. And it's a good, I think, lesson in looking at, kind of carrying through experiments. The in vivo work, I think I would have liked to see more functional experiments with the cells taken out of the germ-free and SPF mice, because it pretty much was like, okay, let's profile, let's adaptively transfer, see what those cells are doing by flow cytometry. I think they do one functional
and
assay.
That functional difference is very minor. It's interferon gamma production.
Right. And they are looking at percentage. So I guess we could say, a 70, whatever this is, 70 to 80% difference. Maybe that is significant functionally. But yeah, I was looking for a little bit more. I was hungry for more.
Yeah, well, actually, they say in the discussion that because there's so few cells in the spleens, we really need to do these experiments with way more mice.
Mm-hmm.
Right.
Way more mice. And one of the things I was going to bring up later, and Vincent, if you want to keep going, I could bring this up later.
Okay.
But, there was a big focus on the spleen. But where does butyrate act? Butyrate acts in the colon, in the gut, specifically the colon. And a lot of the previous literature shows the effects of, I should say short chain fatty acids, we'll get to the butyrate later, on local cells. And so they had the opportunity to tell us more about the colonic migration of these cells, because that to me would be even closer to the short chain fatty acids, but they really relied on the spleen. So I think I just would have liked to see more. I'm guessing they presented the best data they had, so maybe the ileum was kind of not impressive, but I would love to see that local response.
If I remember, when you give mice high fiber diets, you get increases in circulating butyrate. So it does get into the circulation, right?
Yes. People have measured that.
We don't know. Yeah. Yes, but we don't know in their mice, did their diets increase the circulating short chain fatty acids?
They did not test that. That pissed me off.
Yeah, they cited a paper that said it did. Yeah.
Yes, but they did do it.
Yeah,
if they can do all this metabolomics, why couldn't they just run that on a little serum?
For sure.
Just send that out too.
Yeah.
Oh yeah, that's true. That's true. Yeah. That's right. That's true. Okay, so then they do a very simple experiments where they take the germ-free mice and they house them in their SPF facility. So normally the germ-free would be in a germ-free facility to keep them germ-free. If you put them now in the SPF facility, presumably they're going to get colonized, and now this phenotype that they see disappears when you put the germ-free with the SPF mice. Now you no longer have this decrease in the injected cells after, because you've presumably colonized the germ-free mice by co-housing them with SPF mice. All right, so they say microbiota required for this survival of these activated GBT1 cells, right? Activated, they've added the peptide to them before injecting into mice.
That's right.
Okay, so next experiment, they say we want to see the ability of these cells to mount recall responses, memory, right? So they have DCs to which they've been activated with LPS. They've been peptide has been added to them, and those are then injected into mice, both GF and SPF mice, that had previously, 2 weeks earlier, gotten GBT1 cells injected into them, and those are activated. So we're looking for a memory response to the peptide being carried in by these DCs, right? Are their T cells like rapidly going to proliferate?
They should proliferate, right?
Yeah. And so when you do this in the germ-free mice, they say the cells are impaired at generating effectors, secondary effectors, I guess. They're looking at interferon gamma, which would be a CTL-produced cytokine, right? So they're impaired at making these cells compared to these SPF mice. Okay, so there is a impairment in a memory response. And they say, so these antigen-activated CD8 T cells, They go down in number in a microbiota-free environment, and they don't generate memory cells because, well, I don't know if "don't" is the word, but they have they're impaired at generating memory cells because nothing's black and white here, right? It's always less.
Right.
Or slightly
slightly less, yes.
very minorly, true. Slightly less.
Which brings the point, these are valid points that you guys make. This is a good journal, so what's going on?
Mm-hmm.
I thought everything published in cell journals was great.
I know. But, I think the paper, on the whole, did a lot of work. It was good work. I think the back end of the paper I was more convinced of. But it just kind of shows that this is a hot topic right now. So really, because cancer is driving a lot of this interest in short-chain fatty acids, they have shown butyrates have shown to be anti-tumorigenic, especially in the colon. So there's metabolism and immunity is the pairing of that's very hot right now. So you kind of get on that train. It's still good work. I'm not trying to say it's not good, but it's not maybe as robust as I'd like.
Well, I was being sarcastic, of course, because people make housing decisions. Not housing. I don't know what I'm thinking about. People make hiring decision based on whether you have papers in these luxury journals.
That's right. Yes.
But that's not fair. So, you should actually read the paper and not say, "Oh, look, they have 10 cell papers. Let's hire them." Oh, we read them and they're not that compelling, right? Although 10 would be tough to get and not be compelling, but
yeah.
But yet I will say, I am intrigued. It's not that I don't believe this data. I think it's I'm intrigued, but I don't know if I'm convinced.
Well, as you'll see in the end, the whole picture, the whole package here is consistent. And so it's in that sense, it's convincing, true. Right? So if you look at individual things, you can always pick. But I think in the end, there's something going on here with short-chain fatty acids. So
yes,
if it's important, it'll be sorted out, right? That's the way science works.
Hopefully.
So far, we've activated these T cells in vitro. So now they say, well, what about in vivo? If we can activate there, will we see a similar difference between germ-free and SPF mice? So they put these GPT-1 cells without peptide now into mice, and then they infect them with virus. Oh, look at this. There's a virus infection here. Herpes simplex virus 1. And then they take out the spleen and they sort these GPT-1 cells from them, and then they put those into a second set of SPF or germ-free mice. And then they wait 2 weeks and they look for memory phenotypes with these markers that we've mentioned. And they see that in the number of cells with memory phenotypes in the spleens of germ-free mice are lower than in the spleen of SPF mice.
Comparing 0.01% to less than 0.01%.
But Cindy, as you pointed out, if you make a bar graph out of this, right, it looks convincing. And you got nice...
Because they've multiplied it by the number of cells that got in the cell.
But as you said, if you just move the gate a little bit, then that would go away. See? Right. So the bar graphs, if someone is looking at this, wow, the GF is way down there, but it could be different. So I guess.
And I have a question. I wanted to get your opinion. So, they went through the trouble of doing this adaptive transfer of the GB specific cells, and they rationalized, well, we really wanted unrelated to specificity of the receptor, antigen availability, activation status. But I just wonder, these are mice that have never seen HSV. Just infect them, boost them, and see what the memory response is. Do we need this adaptive transfer? Because if you're infecting a naive mouse at the same time, they're all going to be at the same activation status because they were infected at the same time. So it would have been interesting. I don't know if this is specific to
it
that
is a weird
mouse.
way to do it. So, the standard way people did it in the past was you infect, and then you wait till they clear, and then or do a vaccine, right? And then come back and challenge and ask how quickly and how robustly and what types of cells and what types of cytokines they're making.
Because essentially, if the transgenic adaptively transferred T cells are doing this, a normal memory response should do this too. And I just wonder why that wasn't.
And it probably would be as rare a population because you might argue that you want that you want to be able to see the population. So you park a whole bunch of cells in there, but if they're that low, the response you're going to get in a primary mouse who has only a few cells that are specific for that. Yeah, I don't know. I think the benefit of using transgenic T cells is that you can pulse with peptide, and you can look at specific response, and you can use tetramers to identify that particular cell type and specificity. But if you're not taking advantage of that, I'm not sure I see the value.
Mm-hmm. Yeah, good point. Yeah. All right. So far they say these data show that the microbiome are important for making activated CD8 T cells into memory cells. And, with the caveats that you've heard. So next, what is doing this? Because, if you just say it's the microbiome, it doesn't help. It's not mechanistic at all. Mm-hmm. And it's all about mechanism these days, right? If you just don't do mechanism, it's going to be very hard for you to get anywhere. It has to be good mechanism. It can't be mechanism.
Meh.
We talked about meh on TWIV last week.
Yeah, yeah.
So these, of course, as we mentioned earlier, the microbiome synthesizes a lot of things, metabolites, and those are the things that are important. Less what bacteria are present, but what they're making. And so people are interested in metabolomics for that reason. And you can do massive metabolomics analyses of cells and hosts and so forth. So they're interested in these short-chain fatty acids because there's some background that they're made by the microbiota, as we said, by fermentation of fiber. So these short-chain fatty acids are not present in germ-free mice. And I noticed one of the reference for that is Arpaia et al. 2013. Nick Arpaia is here at Columbia. And he was on TWIM a couple of weeks ago with a different story.
Oh, nice.
A different story about...
Oh, is that the engineered bacteria story?
They engineered them to make an antibody against CD, an eat me signal.
Nice.
And they treat tumors with it in mice. Really cool. So then let's see. They feed mice a high fiber diet, right, to see if this is what's defective in generating a CD8 memory response. And they say such an intervention has been shown to cause circulating butyrate levels to rise 100-fold. They don't, as Cindy said, they didn't do it themselves, and it would have been really easy for them to do. Because who knows? Who knows, right? Maybe it's only 10-fold and it makes a difference or nothing. So they transfer these in vitro activated GPT-1 cells into mice, SPF mice, that had been on a high fiber diet for 5 weeks. And then they look at the number of cells that go into memory. And the cells that transition to memory are not different. But when they look at antigen-specific recall with this HSV1, well, they actually infect with HSV1. Then they see a difference between mice on a high-fiber diet and not. So the mice on a high-fiber diet, they had higher numbers of effectors than did in control mice who didn't get a high-fiber diet.
Now, Vincent, would you have liked to see some viral titers, maybe? If this is a truly memory response, are we seeing higher clearance?
Yeah, that would probably be a nice experiment to show that it's functional, right?
Yeah. Well, I mean, they have the mice; they're infecting them. So
that would have been good.
Run some PCR.
Yeah, well, I would do a plaque assay, but I'm a dinosaur. Although I do a lot of visiting of places. In some places, people do plaque assays still, so I'm not the only one.
I did one the other day.
Which virus?
Rotavirus.
Very good. Does it make nice plaques?
Mm-hmm. The coronavirus I worked with previously is better, but it does make pretty good plaques.
Very good.
Thought of you.
Oh, thank you. Someone in the world thought of me.
Everyone thinks
momentarily. That's good. If I've achieved anything in my life, people come into my office, and neither of you have been here, but someday you will.
Yeah,
I have this wall of 6 well plates, right?
Yeah.
And 1,600. Most people don't notice it. They sit here, they'll be here for 2 hours, and then they turn to leave, and they go, "Oh, what's that?" Isn't it funny? You walk into a room, I always look around a room when I go in, right? Because I'm nosy. I want to see what's in people's rooms. A lot of people have champagne bottles, right?
Observant. That's a better word.
Observant, yeah.
Yes.
A lot of people have what?
Champagne bottles.
Oh, yeah, I have some.
So you celebrate something, right?
Yeah, paper.
And your biosafety people don't care about those, because our people would.
I don't
empty bottles in an office? No.
Well, the plaque assay plates, because they were in a BSL-2 put it one time, and I mean, even though they were fixed and everything, but
they have asked me.
Oh, really? Okay.
I'm not sure. Well,
they just asked you if there any.
They
virus
said,
left
"Well,
there?
Look, the thing is treated with 10% TCA.
Yeah.
And then the stain, the crystal violet, is dissolved in methanol-ethanol mixture. So I don't think there would be infectivity. However, poliovirus passes through the stomach, so it could be. So I don't know the answer to that. At ASV, I showed a picture at my keynote. I think, Steph, you were there, right?
Yeah.
You were there as well, Cindy. That's right. Yeah, it's because we did a podcast.
Yep.
And the lady who interviewed me, forgot her name.
From NPR.
Yeah, she said, "Are they clean?" That was her way of saying it, which is funny, right? Are they clean?
That's funny.
All right.
No.
So there might be virus, but I haven't checked. Do you want me to check? I could.
Nah,
Wipe it down.
I wouldn't.
So the thing is, I'm on the IBC.
Oh.
And I'm the virus person on the IBC. So they tend to listen to me, for better or worse.
So then don't even, hey.
I would never put anyone in health in jeopardy. I'm not a mean person. I'm not selfish that I say I need to have this. We need to do this experiment. We don't care about safety. I would never do that. I'm very cognizant of safety. Yeah. So these mice that they fed a high fiber diet, we know there are receptors for short chain fatty acids. And so they do an experiment, which I don't really want to explain, but you guys want to. They do an experiment to say, if we take these away, what happens? So the same experiment that we did, except in mice in which some of their cells, the bone marrow cells in particular, are lacking the genes for these two short chain fatty acid receptors. And they can show that you need these receptors for the high fatty acid diet effect on the generation of memory.
High fiber.
High fiber, yes. Well, we say the wrong thing. As I get older and I say the wrong thing more and more, I'll have to stop podcasting.
No. I mean, it leads to the high short chain fatty acid, right?
Yeah. So, but they didn't directly look at it, right?
So the interesting thing here is the butyrate and these short chain fatty acids have effects on lots of different cells.
Right. Sure.
And so when they do this mixing together, they're using bone marrow from the cells that don't have the receptors for butyrate.
Right.
Those are really for all the different kinds of cells.
That's right.
So the argument is that they mix together wild type and the receptor lacking ones, the bone marrow. So you are comparing it to the wild type, and it's housed in the same mouse. So if presumably some of the bone marrow cells can respond to the butyrate, so the defect would be intrinsic.
So you can, so in other words,
the cell.
You can.
So it's not the perfect experiment, but it's pretty good.
You can compare the T cells with and without these GPCRs.
That's right.
GPRs,
That's right.
Right.
That's right.
And I imagine that if they had so a different type of model where they can do site-specific recombination, we call that Cre-Lox knockout.
Yeah.
I'm assuming they just, didn't have those, and the mixed bone chimera is the best. So maybe we would see a greater reduction in the knockouts if it was, a full knockout. Yeah. Or they could do it, what would be great if and maybe they're working on this now, but if you had a site-specific knockout, you could knock them out specifically in CD8 T cells and then see directly the effect.
And they looked in both blood and spleen in this experiment, right?
Mm-hmm. But in the figure, apparently D and E, they just looked in blood and blood.
Well, it's mislabeled, huh?
There was a lot of
blood,
editing.
That was D and E is at different time points.
Okay, I would.
That spleen
F and G are in the spleen.
Oh,
D. What they did was they parked the cells in, and the bone marrow chimera, and then they ask, are there equivalent numbers of cells very early on, and then they challenge with HSV, and they ask about the recall response, whether or not they are higher or lower. So that's both in the blood, but then the other ones are in the spleen.
my bias was coming through because in figure 1H, they had labeled that as butyrate control instead of, so I was a little biased from figure 1. Look at me. Yep. They did fine in figure 2. My bad.
They did it correctly.
They did.
So next they say, can we add butyrate and see an effect on the differentiation into memory cells? So they again do an in vitro experiment. They take these GPT-1 cells, they add their herpes simplex peptide, and then they add short chain fatty acid to the cells in culture. And then they look at markers or characteristics of memory T cells, which include production of response to IL-15, and IL-15, which I believe will stimulate these T cells, proliferate. Yeah. So they expand, they proliferate. They look at a transcription factor called FOXO1, which is known to be needed for memory T cell differentiation. And they say this pattern is characteristic of memory potential. So they say the butyrate enhances their memory potential by looking at these 3 different things.
And it's a good thing to note that this was only seen when the cells were treated in vitro with butyrate. I don't know if they mentioned exactly here, but if they treat the animals with butyrate, they don't see that same thing. And they do mention that in the discussion that, there are some, it's not a direct effect, and that there's going to be other mediators, because it really is these are interacting with the colon, and then those have to the metabolites are coming out of the colon to the systemic. So, but it's just to keep note that it's in vitro.
Don't go and buy butyrate and drink it.
No.
Oh, I know about this because I was helping somebody write a grant, and so there's prebiotics and probiotics. So prebiotics is this idea of eating high fiber, which provides the substrate for the bacteria that are in your gut to make the butyrate, right? Probiotics are eating the actual bacteria that are capable of fermenting. So enhancing their quantities in your gut can enhance the production of the butyrate. Or you can supplement butyrate. And the thing is butyrate tastes awful.
Yes. It's
very, very unpalatable. So it's not a really good way to treat humans. So you would suggest doing pre or probiotics instead of doing butyrate itself as a therapeutic, unless you're going to deliver it in some kind of pill where you can take it down and then not taste it. But apparently it's not so easy. You can force mice to drink the butyrate. They hate it, but they'll drink it because they don't want to dehydrate and die. So you don't give them a choice and they'll take it. But as far as I understood, the one thing I didn't necessarily agree with, I thought if you supplement butyrate in the mice, it does actually go into the bloodstream. And they argued something about the reason why their oral restoration didn't work was that there were starved cells in the gut that would take up all the butyrate and it wouldn't make it into the circulation. Yeah. But I think that's a little weird because they're not starving these mice of butyrate. They're on a normal diet.
Right. Yep. So
I thought that was a little weird.
Agree. Well, and some of the papers that they cite, I went back and looked, and they were able, especially, I actually think, Vincent, it was the colleague of yours. What is his last name? Starts with an A.
Arpaia.
Arpaia. So he was able to show that peripheral regulatory T cell generation was impacted by butyrate, and their supplementation of butyrate in just a normal animal, I have to go back and see if they're germ-free, it did make a difference. So it did change things. So I agree with that. And again, let's look at the systemic levels. It can make it outside of the colon. You would have to show us more, I think, to make that claim.
Yeah.
So that experiment, adding the butyrate, is cells in vitro. So they say, does this work in mice? So again, you treat the cells and you give them to SPF mice. And you look at memory potential again. This time they infect with HSV1, right? And they say that the treatment butyrate enhances their memory potential, and it doesn't work with acetate or propionate. So again, treat the cells with peptide, then the butyrate, put them in mice, infect, and having the butyrate around helps. Yep. So they say it restores. Remember, these are SPF mice, so they would not have a good memory generation, and so treating them with butyrate improves it.
Yes. Should stimulate with some HSV in these in vitro butyrate treated to see if proliferation is increased. Would have helped.
Yep.
And then now we're moving into another area here.
Get your biochemistry books out.
Yes. They do some RNA sequencing of these GPT-1 cells from SPF and GF mice. And they can identify genes that are differentially expressed when you compare the two mice, right? That's an interesting experiment to do. What's different? And the first step of this analysis, they can see what they call transcriptional signatures associated with memory potential of CD8 T cells. And so these other people have looked and said, what mRNAs are turned on that are associated with memory potential? And they can see some of these, and they can see in the cells, the GPT-1 cells from GF mice, these signatures are lower. And if you add butyrate, they go up. So the butyrate experiment that we talked about, right? Mm-hmm. And these are all taking the cells out of mice. So not only do they have the markers of memory T cells, but the transcriptional signatures associated with them vary in a way that's consistent with the effect of butyrate. Okay, now let's do some metabolism. What's the mechanism? So they say to gain insight into the mechanism. And you need to have mechanism. I would say these big papers, if you don't have mechanism, it's going to be hard to get your work published in them. They use a word called interrogated. We interrogated our RNA sequencing data, which I never liked, but it's in the literature now, and everyone says it's okay. What's wrong with analyzed? We analyzed them. Okay, fine. So they had RNA sequencing. I'm talking to myself mostly.
No.
It's like an old guy complaining.
It's just.
I just think it's a weird. It got introduced in the last few years. I noticed this, and I don't know where this came from. It's like a wayward microbe that is introduced into a colony and proliferates. And then, all right. So they took this RNA sequencing data that I'd already told you about, where they first looked at the signatures of memory, and then they say, what other patterns can we see? And they happen to see one category of gene, which is called oxidative phosphorylation. And this, they say, was the most distinct when they compare the cells, germ-free and SPF mice. Oxidative phosphorylation, of course, which is a way to make energy by oxidizing things. We do that. Bacteria do it. It's very efficient. We inherited it from bacteria because they became our mitochondria.
That's right.
And it has allowed eukaryotes to get very complex. Oxidative phosphorylation. So, you can see all different mRNAs that are involved in this process. And so genes involved in oxyphos are depleted in cells from germ-free mice. And in particular, they see mitochondrial protein complexes. Remember the respiratory chain in mitochondrial membrane, the electron transfer chain. That is part of oxidative phosphorylation. Those genes are down. And they use software packages to do this, right? Because you have all this RNA-seq data, and then you can tell your software to say what's different. And there's one package called Mouse Mitocarta 2.0. I love it. Mice mitocarta to look at mitochondria-associated genes. So that suggests to them that these T cells need to adjust their metabolism to become memory cells, right? And maybe the butyrate and other things are having something to do with metabolic adaptations. And then they begin to measure some of these metabolic activities. They can look at mitochondrial membrane potential and see if that's affected by treatment with butyrate. They again use these GPT-1 cells that you've added the peptide to. And you can see that if you add butyrate, it increases the membrane potential. It's indicative of mitochondrial membrane, that is indicative of increased oxidative phosphorylation. And they can actually measure oxidative phosphorylation. And you can see that you can do that by looking at oxygen consumption. And they see that butyrate increases that.
Now, Cindy, do you have, I think you have some experience with
reading these things?
Yeah. There's the seahorse.
Yep. I was trying to look. I know I mentioned it on a previous episode. We went through this, and I can't remember which one it was. I was going to look that up right now. That's why I was a little slow in answering you, because I was trying to figure that out. But yeah, so this instrumentation that you use to measure this stuff is pretty cool. It's called a seahorse analyzer. And it's one of these things that's super simple, and you wonder, wow, how much money the company makes off of just measuring
that's right. Yes, I remember.
Oxygen consumption. So so it measures two things, and that's it. It's got one probe to measure oxygen, and it's got another probe to measure pH. And by using a variety of different chemical compounds and inhibitors and so forth, you can get a lot of information about how much respiration there is going through the various different parts of the complex, things like spare respiratory capacity, its maximal respiration rate of cells, and also the acidification rate. And these give you a lot of information about fatty acid oxidation, glycolysis, and oxidative phosphorylation.
Cool. Nice. Well, that's what they do, and they say that so if you don't have microbes, the T cells have reduced metabolism compared to either having microbe or butyrate. You get increased metabolism, which is presumably related to the differentiation into memory T cells.
So a lot of my hardcore physiology, nutritional sciences friends, they just giggle at all of this because like if you give a cell a metabolite, it's going to have higher oxygen consumption rate.
Yeah, of course.
And they kind of think it is good. We're looking at the different distinctions of some of the CD8, CD4, all these different cell types. Yeah. But when I talk about this with people who are just in hardcore physiology, they just kind of
sure. Well, it's true. It's true. But of course, if you can associate it with in this case,
disease
memory
or process, right?
I mean, the next step also that they look at is they would probably say, yeah, that makes sense.
Yeah.
So just to remind everyone, the TCA cycle, tricarboxylic acid cycle, is very important for producing energy, and the input is pyruvic acid. And then there's a circle. If you remember your biochemistry, it's a circle. With,
if you don't have nightmares.
It was a circle that you had to memorize, and you forgot promptly after the test with different compounds and converted to one another, and multiple steps, energy can be produced. It's just a wonderful cycle, especially when you put it in some context, right? Because if you just say, memorize this because this is how energy is made, where is it happening and what cells under what you don't learn any of that. But today it's a little more relevant. So pyruvate is the input, and pyruvate is the product of metabolism of glucose. So glucose is taken into cells by specific transporters, and it goes through glycolysis, and that process can generate a number of energy molecules per molecule of glucose. In the end, you have pyruvate, which can enter then the TCA cycle and make even more energy. So they wanted to look at the TCA cycle and the effect of butyrate. So they can what they do is they label these GBT1 cells with C13 glucose.
Like real old school, right?
Yeah,
quite. Very few people
Could be
do
50
this
years ago doing
anymore. I went to a talk yesterday where they're talking about solving the genetic code, and they had to use so much radioactivity that they got entire corridors hot. So they label these cells with C13 glucose, they culture them, and then they look for the incorporation of this label into the intermediates of the TCA cycle. So in the control cells, with nothing added, they have a peptide, I guess, right? Activated, yes. They've added the peptide. They see a lot. The most C13 they see is in citrate, which is the first step in the cycle after acetyl-CoA converts oxaloacetate to citrate. So citrate is the highest, and they say this reflects the entry of glucose-derived intermediates, right? Because pyruvate is entering the cycle and it goes to citrate. In the butyrate-treated cells, you see reduced incorporation of C13 into citrate, and in fact into all of the subsequent components, malate, fumarate, glutamate. And they have a nice figure where they show the cycle in terms of their graphs of incorporation.
With a lot of white space, I still appreciated the way that they set that up. It made a lot of sense.
It's good, yeah.
It was a good figure.
Definitely.
Right.
You could imagine where the cytoplasm is, where the mitochondria is, and how they separate.
So basically, butyrate reduces the input of glycolysis into the TCA cycle, right?
So where's it going?
Yeah. So remember, butyrate increased oxidative phosphorylation. So they're saying, well, it must be somehow rewiring metabolism, mitochondrial metabolism, to utilize not glucose, but something else. Right? What is it? Well, we know that. So I put a picture into the show notes, which is from our virology textbook. It shows the TCA cycle, but also in certain virus-infected cells, the compounds are pulled out of the cycle prematurely before they can go around. So, for example, herpes simplex virus pulls out oxaloacetate to make pyrimidines and purines for genome replication. And cytomegalovirus pulls out fatty acids from citrate to make membranes. And because they're pulling out things in the cycle, you have to add something in somewhere else. And we know that some viruses cause the uptake of glutamine, which can input at the alpha-ketoglutarate stage. So I was very excited when they said glutamine can be catabolized to alpha-ketoglutarate and used to I like this, top up TCA intermediates that promote oxidative phosphorylation. That's exactly right, because if you pull out stuff from the cycle, you're going to lose energy unless you put something in somewhere else, right? So they add C13 labeled glutamine, and they can see incorporation of glutamine into the TCA intermediates in these cells, and it goes up when you add butyrate. So butyrate is changing, they call it a metabolic switch. It uncouples TCA from glycolysis, right? But they add glutamine, and it's called glutaminolysis. They break down glutamine, and that inputs into the TCA cycle. Now, why that happens is not clear. We're not going to learn in this paper.
Yeah,
right?
I think a future effort.
Right, because you may say, okay, so what's the difference of getting your energy from glucose versus glutamine? We don't know. But
specifically for memory response.
Well, at least it's happening in this memory response. It may happen in other things too, right? Who knows?
You're right. Sorry, I guess specifically to butyrate treated cells. These are not cells that had been reactivated, so we can assume they're not memory cells.
Now, even though glutamine is coming in here into the cycle, it's only half of the carbon that goes into the TCA cycle. So they're saying, what else is going in? Because it's not glucose. We know that glucose is not going in. So they say, well, we know that oxidation of fatty acids can provide, is known to be important for the generation of memory T cells. So maybe these fatty acids are supplying the 50% of the carbon in the TCA cycle here. So they do a number of experiments to address that. They show that if you label butyrate now with C13, you can see it's incorporated into the components of the TCA cycle. And this happens in the T cells, that there's been these I forgot the name, GBT1 T cells. They can take up butyrate and use it to make energy through the TCA cycle. The interesting thing here is that it doesn't require the short-chain fatty acid receptors, which were required for the enhancement of the CD8 memory by butyrate, but now they're not important for the input of butyrate into the TCA cycle.
But I think that's because we're only looking. I'm just trying to. So that model, as Cindy mentioned, had knocked out those receptors on many different types of cells.
Yeah.
And so we weren't looking at a CD8 specific. And so I think that makes sense to me, because if this is a more clean system, so now we don't see the same.
So you would say that the effect of butyrate is independent of these short chain fatty acid receptors in T cells?
I think that the story is just more nuanced than if you knock out those receptors. There's a difference specifically to CD8s. I think that there are other cells contributing, and that's why this is not recapitulated here.
Okay.
Well, I also think it's possible that the receptors are signaling receptors, right? And so in response to butyrate, there's those forgot the name of them. They actually signal,
G-protein coupled,
they signal a change in function of the cells. Whereas this is using it metabolically. So if there's a channel or something that can pump it in, you wouldn't necessarily need the signaling channel, the signaling receptors.
That's a good point. Okay. And I think maybe they do bring that up in the discussion too. They talk about some porins that can bring in. So maybe, you're right, that it's just a different mechanism, non-signaling.
All right, last experiment. If you made it to this point, you're good. So
pat yourself on the back.
They look at metabolites. You can do this by mass spec, metabolomics. Yep. So you can say, let's take a sample, and what are all the small molecules that we can detect in it by mass spec? And they look at the spleen here, and they say what metabolites would be available to these CD8 cells. And you can get many, I think they looked at 161. You can put them in different classes, kind of like we did with the RNA-seq. We could say oxidative phosphorylation genes and so forth. And when they do this, they say we can clearly see differences between the GF and the SPF mice. 75 differentially regulated metabolites. Okay, that's nice. What are they? Well, you could go look at them, but they don't really comment on that anymore. But presumably that they're going to work on that. Well, this is actually a problem with these massive data experiments, is that often they're published and no one ever follows up. Even the lab working on them, they go into something else, which is kind of not really, what should I say? It's a different kind of science where instead of, in this paper, they did some very nicely designed experiments to address questions, but then you get this huge metabolomics thing and you have all these data and it doesn't tell you anything. It just tells you that there are differences.
Right.
Anyway, so they also can see differences in glutamate metabolism, oxidation of long-chain fatty acids. So these metabolites are changing depending on whether we're looking at GF and SPF mice, which is consistent with their model, but they don't say anything beyond them. But presumably you could mine that a little more carefully and design some experiments to address hypotheses that are generated by these metabolomics data. So that's what we have. We have this observation that this CD8 T cell memory seems to be dependent on microbiome, which in part is because it's butyrate that's important, which is a fermentation product of fiber, diet fiber. The butyrate seems to have an effect on metabolism, where it changes the oxidative phosphorylation from using the products of glucose to using glutamine and butyrate itself, which is very interesting. So the implication is that butyrate is being plugged into this TCA cycle, and somehow that's affecting the generation of memory CD8 cells. And that's the big gap there. What's going on, right? That's what we want to know, the mechanism. But it's not just butyrate, as they say. There are others. And they cite some really interesting data where it's been shown, for example, a recent report showing CD8 positive T cell responses in the intestine required 11 commensal bacteria to act together. And the butyrate doesn't always work well by itself. You have to have other commensals that are making other things. So it's not as simple as just butyrate.
Right. Yeah. And there's a couple, when I was reading up, people trying to, because they realize the benefit of butyrate and trying to design, probiotics or treatments that can enhance butyrate. And it's also hard because, you have these resistant polysaccharides that are degraded by what are considered primary degraders, which are Bifidobacterium, Ruminococcus, but they produce intermediate products, and those intermediate products are then broken down and by a different subset of bacteria to produce butyrate. So it does make it challenging when you consider there's degraders and then butyrate producers and many different types of bacteria that do this.
Mm-hmm.
So I just want to mention two things from the discussion. The last paragraph has two kind of broadly focused points. The first is if you're trying to design a T-cell based vaccine, right, against the virus or bacterium, which would not induce necessarily antibodies, but protective T cells, you're going to have to worry about diet, right? Because maybe if you're, this is very interesting. If your study population has a poor fiber diet, maybe that will impact the generation of T cells. So that's another variable in a vaccine trial. What are they eating?
I wonder if people have looked at people who are on paleo or keto diets compared to
that's a good question.
Higher
that would be
carb fiber diets and see whether there's a difference in response to vaccines.
Yeah, or even Western versus non-Western because we eat less fiber.
Yep.
In vaccine responses.
So when we talk about studies, epidemiological studies or vaccine trials, and we talk about confounding effects that weren't considered, that's one. Diet. If you don't ask people what they're eating, could have a huge effect, but you don't sort by it. So that's a good point. And the other is, if you're doing T cell therapies, right? CAR T cells. If you, and part of that is to make memory. Then we got to consider the diet. Maybe a clinical trial needs to be done to see the efficacy of these therapies based on a high fiber diet or not. And then if that really helps, then you have to say, oh, and by the way, after we give you these T cells, you better eat a lot of fiber. I think those are two kind of practical outcomes.
Or at least we know, I think what we can say about the in vivo work is that pre-treatment of T cells you're going to transfer, pre-treatment with butyrate does impact the memory development.
Yeah.
So you could pre-treat butyrate
CAR T cells. Yeah, that would be interesting. Right. That would be a good trial, right? All right. Well, it's an interesting paper. It's provoking some thoughts, which is good, and certainly more experiments. And I like getting past just we need bacteria to a mechanism. So that's why I like it.
For sure.
All
Definitely.
Right. There you go.
Nice paper. Good paper.
Yeah.
I think it's interesting. Okay, we have a couple of emails here. Let's just go through those. Steph, you want to take that first one?
Yeah, sure. Hello, Immune. I'm working back on the episodes and just listened to episode 22. I was wondering if there was a father's version of the book on motherhood in the lab. I am getting married soon. I plan to go to graduate school. We like to have children soon. I wonder how new fathers do in their first years of a PhD program. Yeah, I want to, this is sincerely David. I want to apologize. I know I have been mother-centric when talking about it. I should just say parents because, it does, I don't think the paternity leaves are I think some universities have them, maybe most, but I don't know if socially it's as well accepted. So that's definitely something to consider. Vincent, did you have kids during the first years of your PhD program?
No, I didn't.
Okay.
I had kids during not even the first years of my assistant professorship. Let's see, when I had our first kid was in Good. I can't remember our first kid's birth. 1994. So I arrived here in 1982. I was already here 12 years.
Wow.
And my wife was already established at Merck. Yeah. So we had kids later, but I know people who had kids as soon as they became assistant professors. I knew people who had kids as graduate students. The whole gamut.
I would imagine that at any time it's going to be challenging and rewarding. And I think as long as, you're working with a mentor who's, somebody who will work with you I think it will be fine. I don't see,
everyone has to work out their own situation. And you can't tell people what to do based on your experience because it's your experience.
Right.
People used to tell me, oh, you should have kids. They're so great. And I said, I'm supposed to believe you on this? And this is a life-changing thing. And so after the first kid, I said, wow, they were right, but I wouldn't have known it until, right?
It's true.
And for some people, it's not so great, right? So everyone's got to sort out their issues. And if I've learned anything in life, don't tell people what to do. You can suggest and give advice, but you can't tell people what to do. All right. Cindy, can you take the next one?
Oh, sure. Alexandra writes, "Hello, Immune Team. I'm a first-year MSTP student at UCSD and an enthusiastic listener of the Immune podcasts. I listened to your Immune 22, Engineering B Cells episode. What was the phase 1 clinical trial that was briefly mentioned at the beginning of the podcast? I think it was based off of some of David Baltimore's work. I'm always curious to track the translation of these types of therapies into the clinic and would love some more information on it. Thank you, Alex."
Okay, so the clinical, I just looked it up at clinicaltrials.gov. I searched for AIDS, AAV, and antibody because that's what they were doing. They were giving a broadly neutralizing antibody gene in an AAV, adenovirus-associated virus vector. And the study is called VRC 603, phase 1 dose escalation study of safety of this virus. It's apparently still recruiting. I don't know why it's recruiting because it started in 2017. It's weird. I think it's just not been updated, but I'll put the link in for you, Alex, and you can check that out. But clinicaltrials.gov is really great for finding ongoing clinical trials of all sorts. So this is a safety test. It's to see, if you produce a lot of antibody, a single antibody from a vector that's going to last a long time in the person, is that a problem? Now, I don't know if it is a problem, what they do. I don't know if there's any way, I don't know if they have a kill switch in these vectors, where you could add something to turn it off.
Yeah, we've talked about that when we talked about the CAR T.
So they're going to just take people's temperatures. They're going to get...
pretty crude.
Yeah. So there you go. That's that. Okay.
Yeah.
And then we have one from Ryan who writes, "Have you seen this article on Wakefield?" So he sends a couple of
Three
links.
different ones.
3 different articles about
I looked at that.
Wakefield and Elle Macpherson. I don't know what the connection is. Oh, I guess she's dating him.
They're dating. Yeah, I think that's the implication of the article.
Well, Elle, I'm sorry you don't have good taste.
That's crazy. I did not know that.
Yeah, I don't know.
From the Daily Mail.
Just, mind-boggling. Wow. But, yep. Okay.
People can do what they want, right?
They can.
They can do what they want. And he has made a lot of money off his shtick. So
I think she probably has a lot more money.
And you're right. She probably does have a lot more.
Ah, boy.
Money isn't everything.
And you know what? His shtick is telling people not to be vaccinated, which is a terrible way to make money. But
yeah, so we're talking, if those of you who aren't knowing who we're talking about, this is Andrew Wakefield. And so he's really notorious, I would say, for spearheading a very unethical study it was later found to be unethical, on trying to demonstrate a correlation between certain diseases and the measles, mumps, and rubella vaccine. And the problem was he was making his own vaccine on the side, and he paid parents to have their children be in this. There was selective choosing of the children that were going to be in this trial. The whole thing blew up. The amazing thing is that in that paper, he said there was no connection with autism. Now that's his whole thing, saying that the MMR vaccine was causing these serious problems. And so it's just a big old mess out there. But he is the champion of anti-vaxxers and really supports everything that has to do with this now. And I think it's mostly just making money and fame off of it, which is really quite sad.
So Ryan writes, "This is why we have the measles scares in various parts of the U.S., and Wakefield staff members are setting up campaign offices all over the U.S. to scare politicians in other states and Washington, D.C. to not use California's vaccine laws in the 2020 elections. Wakefield just got called out for living a luxurious lifestyle." Yeah, I'm afraid this is going to be a campaign issue. One party will say, "We won't make you get vaccinated," and the other party will probably not say anything, but it's terrible.
Well, if there's any silver lining to this whole thing, the number of individuals who are actually anti-vax are a minority. They're loud and obnoxious in a lot of cases in the way they interact with people who don't believe the same things as they do.
Yeah.
But they are the minority. The vast majority of the people believe in the science and they vaccinate their children.
Yeah, so sometimes the minority can have a big sway, as you know.
Influence. Yes, they can. That's true.
Yep. All right. Let us have some picks of the week to wrap up this immune. Steph, what do you have for us?
Yeah, so I'm doing a little cross promoting here.
For you.
Vincent has another podcast. I don't. How many does this make it now?
Well, I don't include Urban Ag anymore because that's defunct. So this would be 6, I guess.
Okay, so the 6th podcast is This Week in Neuroscience, and the hosts, including Vincent, are Ori Lieberman, Aaron Calipari, and Jason Shepard, and I'm going to listen to it on my drive home. It just popped up.
Great.
Yeah,
these people are good card-carrying neuroscientists. So I'll learn a lot from them, and they work in different areas, so that should be fun. And the first episode, they basically talk about how they got into neuroscience and what they work on. But the first few episodes, their plan is to do some papers that will explain the fundamentals of the field, basically, to try and teach people what it's all about before we get into other research papers. So I'm looking forward to that. Nice. It'll be monthly, yeah.
So which is harder, do you think, or which will be harder, immunology or neuroscience for you?
Probably neuroscience.
Yeah.
Because I've had some contact with immunology over the years, right?
Right.
Through my colleagues and seminars and meetings and just general interest. But I've had much less contact with neuroscience, right? So I think it'll be harder. Mm-hmm. The name of this first episode is called The Ultimate Challenge, and that is a quote from Eric Kandel, who said, neuroscience is the ultimate challenge in biology. To learn how memory and emotions and all this that works is really hard.
Yep.
So it may not even be possible. Who knows? But these individuals are really good. They were a lot of fun to do that first episode, and they're going to carry this forward. I'm just kind of a facilitator, and they said you'll be the person we're trying to reach who doesn't know any neuroscience, and that's
great.
That's fine.
That's sort of how you ran some of the other ones too, like microbiology. Well, not really, but yeah, parasitology.
Well, certainly parasitology.
Always say I'm like the one who doesn't know anything.
Yeah, I ask questions like, well, what does this mean? How does this work? And they're fine questions to ask. So I can't wait to say, all right, what's a patch clamp? What do you do with it?
Good. Yeah, I think I would want to know that too.
Thank you. Well,
and I'll tell you what. So this is a huge deal, having this twin coming out. Okay, because for those of you who are listening, and Steph, you could speak to this. We don't do a lot of work for this podcast, but we put in our efforts to make sure that we're reading the papers and have some good questions and some good discussion. But Vincent, the amount of work that you do for these things and having 6 different ones that you're doing weekly or monthly, this is really impressive. I mean, it really is amazing. And so shout out to you from all the listeners, because you really deserve a lot of credit for this. You really maintain this whole ship. It's amazing enterprise. Yeah.
Thank you.
It's fantastic.
I wouldn't be able to do it without all of you hosts. So, right? I could do one podcast by myself, which would be but it wouldn't be any fun. But I always say the hosts of all of these are amazing because they agree to do it over and over again. And I kind of just, I'm a conductor, right? And that's a
great way to look at it.
But I appreciate it, but I think it's crazy in a way for me to be doing so much because I'm a professor. I'm not supposed to do this.
Are you repeating what your department head has said?
I'm not supposed to do this, but I'm not going to stop. Okay, because I think people like it, and I will do this forever. So,
you know what I think we can do.
But I have guilt. Let's just say I have guilt.
Yeah. Well, I was going to say, I think one of the things that we can do as co-hosts is try to promote more, to get people to donate more so that maybe you can get a person to take some of the production. Because I think, if something were to happen and you couldn't do this, we don't know how to record and edit and post. So I think what we can do is promote more, hopefully get more money so that you can have help.
Yeah, I worry about the fact that I'm doing all the tech. I don't like that because I've been lucky that I've been 10 years. I have been healthy, right? And I should either teach some of you how to do this or get someone. You should
teach us. Yeah, we would learn.
Yeah, I could easily do that. And especially with this way we're recording now, it's not as hard. So,
right.
Okay. Thanks, Steph, for shouting that out. Cindy, what do you have?
So I put a link to an article that's in Science, Translational Medicine, and it's about graduate school and being a graduate school, and it's called Graduate Abuse. And I don't read a lot of it, but basically it's saying like, does your boss, your PhD advisor or postdoc advisor expect a lot of you, and are they pushing you? It can be hard, but that's not unreasonable. But are you being threatened or screamed or insulted? That's right. Drawing a line. And I think it just raises awareness of the mental health of students, because I think I see a lot of students struggling. And I don't know if it's just that it's more acceptable to talk about it now, and so there's less hiding of it, and so it's a lot more in your face, and you see the students struggling and they talk about it more. Or if there really is just, things going on in the society and the way people are raised that they just have a harder time. And I think it's good to be aware of it as advisors, because, you're going to have students who come to you and they're struggling. And I think a lot of us are ill prepared to handle that. And so I think just, raising awareness for this and knowing that it's a challenging time to be in graduate school and pursuing a PhD, which is hard enough. And if you have a bad advisor, sometimes you need to make a change. I had a poor experience in graduate school, and I switched PI advisors, and it all turned out great in the end. But while I was under that, I thought it was me. And a lot of times you think it's you. I screwed up the experiment. I must be the only one who ever does that. And that's just not true. And so just, being aware of that, if you have a chance to read that article, it just, lets you know that there are certain things that you should be aware of that just aren't okay. If you're in a lab and it feels uncomfortable, it might be a good reason why.
It's good. Yeah. You should
awareness.
It's short enough. You can read it in 5 minutes.
Yeah, it's really fast.
Yeah. And I used to read Derek Lowe. He used to have a column called In the Pipeline, which now has apparently moved to Science Translational Medicine.
Oh, okay.
But it was a really interesting column about the drug pipeline for pharma, right? And and he is a person in pharma, and so he had wonderful insights. And so he's a good writer. Cool. And my pick is a New York Times article. It's by Don McNeil. Yes. It's titled, Scientists Were Hunting for the Next Ebola. Now the U.S. has cut off their funding. So I didn't know this, but there was a government research program called PREDICT, which funded pathogen discovery globally. It's about 10 years old, and over those 10 years, it's $200 million has been invested by a number of different people who have collected samples. They've found over 1,000 new viruses. They've trained 5,000 people in 30 African and Asian countries, built or strengthened many medical research labs, mostly in poor countries. So the point is to see what's out there, and that maybe it helps us to be ready if there's a new pandemic, right? And the Bush administration, Obama administration supported it. Yeah, the current administration has canceled it.
Unbelievable.
Right? I don't know why, but I think this is a cheap, like someone says here, the last Ebola outbreak cost $5 billion. And this is $200 million, and this is a bargain. And we really should have kept this up, and it's really unfortunate because it's cheap, and it's too bad. I'm just sorry that things like this are being cut, and there's no good reason. All right. Let's give away a copy of a book.
Yay.
It's very heavy. It's big.
It sounds like it was.
8th edition, Manual of Molecular and Clinical Laboratory Immunology. Published by ASM Press. And, it's just really Section A, General Methods. Section B, Immunoglobulin Methods. Section C, Complement Methods. Ooh, look at this. Section D, which is many, many chapters, flow cytometry.
Oh, we talked about it earlier.
Tons, very many. And then
learn how to
functional cellular assays, cytokines, chemokines, immunohistology, immunopathology, on and on. And then it's broken down into viral, bacterial, fungal, immunodeficiencies, cancer, organ diseases, transplantation, laboratory management, only 2 chapters.
Wow.
So this is over 1,200 pages. So we're going to give this away because I should probably keep it and read about flow cytometry, right? But I'd like our listeners to have it. So just send an email to immune@microbe.tv, subject line manual, MANUAL. That's easy, right? If you want to write something, that's great too, because we love all of you.
And we love to hear your emails and questions.
We want to interact with all of you.
I just saw this movie, Bohemian Rhapsody.
Mm-hmm. Oh, you did?
Yeah.
Did you like it?
It was good. I'm not a movie person, but my daughter wanted to watch it with us, which is unusual that she wants to do anything with us. So I thought it was a good thing. Maybe I should say yes. So I'd like to be, and because the music is good. I've always liked Queen's music, but there's this point where they're touring everywhere, and wherever they go, "We love you, Detroit. We love you," or New Orleans. So I thought we should say that in our podcast. We love you, listeners.
We love you, listeners.
We love you, listeners.
But the audience loved it when they said that. So we love you, and we want to give you this book. Send an email to immune@microbe.tv.
Need a cheer. Yay!
And, of course, immune@microbe.tv is also where you'd send us questions and comments. And if you like what we do, consider supporting us. Microbe.tv/contribute. We have ways where you can use PayPal or Patreon. You could give $1 a month if you'd like. It's not very much. We know that many of you don't have a lot of income. But this is a worthy cause because we have a lot of people disseminating knowledge for free. They don't get paid to do this. So I would love to have some support, not only for our expenses, which we do have, but eventually to hire someone to help me. It would be nice. I don't want to take any of the money myself. I just would like to hire even a part-time person to help out. So microbe.tv/contribute. And if you're a supporter, we answer your questions first, as always. Okay, that is Immune 25. Today we've had Cindy Leifer. She's at Cornell University. Over on Twitter, she's Cindy Leifer. Thanks, Cindy.
Thank you.
And Steph Langel is at Duke University. At Twitter, she is Stephanie Langel. Thank you, Steph.
Yeah, thanks. This is great.
Vincent Racaniello. You can find me at virology.ws. I'm on Twitter also, PROFERR. Music you hear at the beginning and end is by Steve Neal, stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.